Okay, maybe we can start. So welcome everyone for this weekly webinar, SEM. So today we are going to listen to Laura Galloway, who's talking from the US. So it's the morning for you, Laura, I think, Friday afternoon for us. So thank you everyone for coming, even though it's not the usual time. Just a quick reminder, I think you are starting to be used to it, but you can listen to the talk and in the meantime also type your questions in the Q&A window that you will find at the bottom of the screen. You can also vote for any questions if you feel they are relevant, and that will rank the questions according to their votes. And at the end, we will try to make it as interactive as possible by opening your microphone so that you can ask your question and ask for further details. Okay. Right. So today we are welcoming Laura Galloway. Laura did her PhD in population biology at the University of California in Davis, and then conducted a postdoc research in quantitative genetics at the University of Maryland. Since 1996, she's a professor at the University of Virginia in Charlottesville in the US. And her research interests are currently focusing on ranging from mating system evolution to the evolution of reproductive isolation and the evolutionary consequences of polyploidy. In her lab, she uses ecological and genetical approaches to explore all these mechanisms of adaptation and the patterns of evolutionary change in natural plant populations. So today she will talk to us about how plant mating system evolution can be affected by both contemporary pollinators and historical processes across the species range. Right, so with that, I will start listening to you, Laura. Thanks very much for being here in the morning, and yeah.
Wonderful, thank you so much. You can hear me okay?
Yeah,
okay,
perfect.
Good. It's a real honor to be here and a pleasure to get to talk to you all. Of course, I wish I was there in person, but in the world that we live in now, this sort of seminar is possible. So I really appreciate getting to talk to you. Today, I'm going to talk to you about work that we've been doing to understand the extent to which trait evolution is influenced by local selective environments and by historical processes. So, as we all know, climates are warming, and in response to this warming, plants and animals have changed their distribution. So this figure shows results from species richness on mountaintops in Europe. And what you can see is that this has increased over time. And this is because as the mountaintops are warmer, lower elevation plants and animals are moving up to the mountaintops. So they are expanding their ranges into newly suitable habitats. Now, climate change is not only occurring now, but has also occurred many times over evolutionary history, albeit not quite as rapidly. So if we look at the last major glaciation in North America, this occurred somewhere between 18 and 20,000 years ago. And at this time, the plants and animals were mostly located right down here along the southern coast. As the glaciers receded and climates warmed, you have plants spreading out of these refugia north slowly over time until they inhabit their current distributions. Now, this process of range expansion may shape the genetics of populations. And this is because when ranges expand, what happens is you have a portion of individuals within the glacial refugia that colonizes and forms a new population. and it brings with them a sample of the genetic variation in this refugia. And this process happens over and over again, such that by the time we get to range margins, we find that there has been a reduction in overall genetic diversity simply due to these founder effects. In particular, there is a loss of rare alleles, and the drift associated with founder effects and small population sizes may lead to an increase in frequency of different alleles in different populations, sometimes an increase in frequency of mildly deleterious alleles. Okay, so then this shaping of genetic diversity due to range expansion is expected to affect the evolutionary potential of traits, and it may contribute to geographic variation in trait expression. Alternatively, patterns of geographic variation in trait expression may be due to selection associated with local ecological variation, or as I say here, variation in contemporary, that is current day environments. An example of this would be something like under the abundant center model, we expect populations in the center to be common and large, whereas those at the range edges, populations are going to be few and far between, since that is the definition of a range edge. These few populations may be small. For a plant, they may be so small that either mates are limited or they're not very attractive to pollinators, so pollination is limited, giving rise to overall not enough pollen received by flowers to set all the seed they could. These pollen limited condition then may select for traits related to mating system, in particular, an increase in selfing. So in addition to these sort of biotic selective environments, there may be abiotic selective environments that vary across a geographical scale. For example, there's temperature gradients over many species ranges that would result in selection for heat tolerance at southern parts of the range and cold tolerance at northern parts of the range. Okay, so this then is a little bit about how historical factors and contemporary environments may affect trait evolution. And today I'm going to talk to you about mating system variation. So mating system in plants, as many of you know, is the tendency to reproduce either via outcrossing or selfing. Outcrossing is ancestral, and selfing has evolved many times in many different species, such that while about half of all plants, I should say angiosperms, are mostly or fully outcrossing, 30% have intermediate rates, and 20% reproduce almost exclusively through selfing. Okay, so there is a lot of variation in mating system. The question is, why do we find this variation? Because outcrossing is ancestral, we typically study mating system variation as factors that favor a transition to selfing. And we interpret this in terms of the benefits of selfing and the costs. Okay, so the most important benefit of selfing is reproductive assurance, and this is the ability to reproduce without either mates or pollinators. Not surprising. The biggest impediment to the evolution of selfing is inbreeding depression. And this is a reduction in fitness due to the expression of deleterious alleles associated with the increase in homozygosity that occurs with inbreeding. All right, so you've got these deleterious recessive alleles, they're expressed, we get inbreeding depression. So these benefits and costs can vary depending on both genetic and ecological conditions. So as a consequence of this variation, we find variation in mating system among populations of a species as well as among species. A very common pattern is to find an increase in selfing at range edges. This is manifest as a loss of selfing compatibility, finding traits that are more permissive to selfing, so allow selfing to happen more readily, as well as direct evidence of just increased selfing rates at range edges. So the question becomes, why this geographic pattern? Well, based on what I've said, as you might expect, one hypothesis for why there's more selfing at range edges, in particular the leading edge, that is the edge furthest from the glacial refugia, is that range edges have a reduction in genetic diversity. With less genetic diversity, there are simply fewer recessive deleterious alleles, such that the inbreeding depression as a consequence of selfing is smaller. With smaller inbreeding depression, the cost of evolving selfing goes down. So it's more easy for it to evolve. Alternatively, there may be more selfing at range edges, as I suggested earlier, because populations at range edges, by virtue of being small, few and far between, may experience more pollen limitation, which favors the evolution of selfing as a mechanism of reproductive assurance. Okay, so both of these are different mechanisms for why you might find greater selfing at range edges. And what I'm going to do today is use mating system as a mechanism of evaluating these two different contributions to trait evolution. And I'm going to do it using this species, Campanula americana, or American bellflower, as a study system. American bellflower is a monocarpic herb growing as both annuals and biennials. As you can see from these pictures, it is insect pollinated. It is largely outcrossing, however, fully self-compatible. So this species of Campanula is found in the eastern US. This map was made using an ecological niche model or a spatial distribution model where warmer colors indicate more suitable habitats. And then I've just sort of freehand drawn the range boundary. And so you can see that, not surprisingly, there's a good match between the suitable habitats and the current distribution. Okay, so I've talked about this as a mixed mating species, and given what I've said, you probably expect that there's variation in mating system. How do we determine this? And what do I mean by mating system? So we went out into the Western clade, and I'll get to that in a couple slides, and we selected 24 populations that are going to be focal populations for most of what I talked to you about for the rest of the seminar. We took seeds from these populations, we grew them up in the greenhouse, and we simply scored the proportion of flowers on each plant that became fruits without pollinators. And this is something called autonomous selfing, or the ability to self without a pollinator. You can see an example of that here. We've got a node. These two flowers have become fruits. This one and this one haven't. So if we were just looking here, we'd say 50% autonomous selfing rate or selfing potential. Okay, so we did that across plants from all these populations. And what we found is huge variation in selfing potential. Some plants, none of the flowers became fruits. Other plants, they all did. And what I did next was calculated population means, and I'm showing that to you on this figure here. And you can see pretty easily that there is a geographic pattern to selfing ability, with redder populations, those with higher selfing rates, up here in the north and the west. So red is about 80% of flowers becoming fruits automatically. And then here in the east and the south, lower numbers, yellow is about 30% converting to fruits without pollinators. So why this geographic pattern to selfing? Well, the first thing to ask is whether this mating system variation shows a signature of historic range expansion. In order to answer this, the first thing we need to know is what is the pattern of range expansion. And so to address this, we can use a phylogeographic approach, and that is to infer range expansion from a phylogeny. So here's a phylogeny made using chloroplast DNA. And what you can see is that there are three main clades, this one, and this one. And today I am only going to talk about this upper clade, the Western clade. And that's because this is the only clade, or this is the clade that has the greatest amount of mating system variation. And the basal population in this clade is this haplotype that is white colored. And what you can see is that this white haplotype gives rise to two branches, right? The yellow-green branch, and that is here, largely but not completely west of the Mississippi. And then this blue-pink branch, and this is largely east of the Mississippi, and these represent the other clades. The white haplotype, the most basal, is, as you might expect, for where we would predict glacial refugia to be, and that is along the southern coast. But you can also see that it occurs in the southern Appalachian Mountains. And this is important for the next part. Okay, so this phylogeny was made using chloroplast DNA. We also wanted to make a phylogeny using nuclear DNA because it can give us a more nuanced understanding of range expansion, simply because there's more variation present. So we conducted rad-seq analysis and explored patterns of nuclear variation using this whole genome data. And I'm about to show you results that look at the spatial pattern of derived alleles in site frequency spectra. And that is shown right here, where the cooler colors, the greens, indicate more derived alleles, and the reds indicate more ancestral alleles. And I should note, here's the Mississippi River. Remember, we had the sort of eastern and western clades from the chloroplast DNA, and then this is the blue is the extent of the last glaciation. So what this tells us is, for these 24 populations, this X marks the spot in eastern Kentucky has the greatest concentration of ancestral alleles, and therefore is predicted to be the location of the glacial, or the last glacial refugia for these 24 populations. Okay, so this next thing we did with this nuclear data is bearing in mind that the chloroplast data gave us a strong east-west split. And if we look at the nuclear data, various analyses we did also gave us this east-west division. This happens to be the results of a structure analysis, with one group coming out here in yellow and the other in red. You can see this is largely east-west, largely divided by the Mississippi River. So when we saw there was this strong east-west pattern in the data, we tested some additional models of range expansion. So the initial model I showed you tested whether all populations were derived from a refugia in eastern Kentucky. Then we tested two other populations. The first one is that the eastern and the western clusters were derived from separate refugia. And then the third model was that the eastern cluster, the red populations, were derived from one refugia, that they colonized a western refugia that gave rise to the yellow populations. So we evaluated how well each of these three models fit the data. Again, using this analysis that looks at the spatial distribution of derived alleles in site frequency spectra. And what we found was that this nested refugia was the best fit model. Nested in the sense that the second refugia came from expansion from the first refugia. So for the rest of my talk today, I am going to talk about colonization distance as the distance between each population and the refugia. So for the red population, this is the distance simply back to this refugia. For the yellow population, it is the distance to the western refugia plus the distance to this eastern refugia. I should be saying refugium. Okay. So that's the pattern of range expansion. Does it affect genetic diversity? Here are two classic estimators of genetic diversity. And what you can see is that there is a very strong match between colonization distance and genetic diversity, with here about 61% of the variation in genetic diversity explained by colonization distance. Over on this right one, we have Tajima's D. Larger, again, very strong fit to colonization distance, where larger values in Tajima's D tells us that there's a scarcity of rare alleles and a history of reduced population size. So both of these estimators of genetic variation then tell us that this post-glacial colonization has resulted in drift, small population size, loss of genetic diversity. Okay, so that is with neutral markers. Next, we were curious, okay, drift has happened. We see it in population genetic structure. Do we find that it is associated with drift load? What is drift load? Drift load is a decrease in fitness due to the fixation of mildly deleterious alleles through just random processes. And drift load is typically estimated by crossing populations and looking for signatures of hybrid vigor. Okay, so we took our 24 populations, grew them up in the greenhouse, conducted within and between population crosses, and then grew the offspring, measured a number of traits. And today I'm only going to talk to you about cumulative fitness. And what did we find? What you can see is that there is a clear positive relationship between colonization distance and drift load. And this is drift load for cumulative fitness, such that the further populations are from glacial refugia, the greater the drift load. So the more hybrid vigor in combining them. Okay, So again, we have a signature of genetic drift that has affected both neutral and expressed genetic variation. The question is, what about mating system? Do we also see a signature of this historic range expansion on mating system? Given that I showed you this map earlier, you probably already know the answer is yes. And I just wanted to show you the figure here. As you can see pretty clearly, there is a nice positive relationship between colonization distance and selfing ability. Why? Where is that coming from? Well, what we predicted was that the reduced genetic diversity associated with colonization would lead to a reduction in deep inbreeding depression, which would then create conditions where it's easier for selfing to evolve. So we wanted to evaluate levels of inbreeding depression in these 24 populations. So we did within population crosses, self crosses, again, for all these populations, took the seeds, grew them up in the greenhouse, and I'm going to talk about cumulative fitness here. And our predictions are long colonization distances, little inbreeding depression, short colonization distances, lots of inbreeding depression. Well, I'm just going to tell you, that's not what we found. We found there was no relationship between inbreeding depression and colonization distance. Was there variation in inbreeding depression? Yes. In this population, there was an 80, maybe 75% reduction in inbreeding depression of selves relative to outcrosses. In this population, it was only maybe 25%. But look, the colonization distance is about the same for these two. So there is absolutely inbreeding depression, but there's no geographic structure to it. Okay, so there is a signature of historic range expansion for selfing ability, but not for inbreeding depression. So again, we're left with, why do we see this geographic pattern in selfing ability? And what I'm going to do now is turn to these alternative hypotheses. The idea that mating system variation may be driven by contemporary ecological factors. In particular, might pollen limitation have driven the increase in selfing that we see across the range? Few pollinators, few mates at range edges lead to select for an evolution of selfing. Okay, in order to do this, figure this out, we needed to do some field work. And I'll say this was a huge project. We went to these 24 populations multiple times in each of 2 years, and I wanted to introduce the major players, in particular Matt Koski, who is a postdoc with me on all of this work and now has a faculty position at Clemson University. Also, my collaborator Jeremiah Bush did all this with me. And then for this field work, we were joined by Dina Grossenbacher and Liao Kuo. And then just briefly on the pollinators, we've got bumblebees, a specialist megachilid campanulae, and then halictids. These latter two not only pollinate, but they also eat a fair bit of pollen. Okay, so for all this field work, I'm only going to talk to you today about pollen limitation. So pollen limitation, how do we measure that? It's pretty straightforward. We mark sunflowers on plants, don't touch them, they're controls. On others, we add extra pollen. If there are more seeds in those that we add extra pollen to than the control, we'll know that plants weren't receiving enough pollen from bee visits to make all the pollen they could. In other words, their reproduction was pollen limited. Okay, and I'm just going to simply plot pollen limitation against autonomous fruit set. You might expect here that large levels of pollen limitation would select for increased ability to self. So we're expecting to find populations up in this corner. So this kind of relationship. What do we find? Oh, there it is. What do we find? We find lots of variation in pollen limitation from none to producing only about half the seeds they could, no geographic pattern. Okay, so if it's not pollen limitation that's driving the ability to self, what could it be? We decided next to look at outcrossing rate, so took seeds from these natural populations, grew up progeny arrays, and estimated outcrossing using microsatellites. And I will just jump right to the results here. And you can see pretty clearly that there is no pattern at all. We might have thought that plants that could self would outcross less, but that's not true. Or where pollen is limiting, there would be more selfing and less outcrossing. That's not true. All right, so at this point we were kind of discouraged, right? We put in a huge amount of work to get this sort of contemporary ecological information on mating system, and nothing is related to selfing ability. At this point, an undergraduate project actually helped us see a little bit of pattern. Leah Liebman was working with me for the summer, and she was taking populations that had high selfing ability and those that had lower selfing ability, constructing them into their own artificial arrays and placing those arrays under conditions where there were few pollinators or lots of pollinators, and then looking at pollen limitation and outcrossing rate. Because her results turned out to be interesting, we took our data from natural populations and did the same thing. Divided those natural populations into those with lots of selfing potential, those with less, and then looked at the relationship between pollen limitation and outcrossing rate. Okay, so with that buildup, what did we find? This figure here is for the experimental arrays. This is for natural populations. And let's start by focusing on high autonomy populations, those that have high rates of potential for selfing. And what you can see is these show a nice tight negative relationship between pollen limitation and outcrossing rate. So if pollen is limiting, they self. If they are receiving lots of pollen, they outcross. So we talk about these high autonomy populations as having a flexible mating system that is flexible to the amount of pollen they receive. Same is true for natural populations. These lower autonomy populations, the open circles, show no response to pollen limitation. So overall, these lines are pretty much flat, and a whole lot more variation in outcrossing rate. So then we found from this sort of set of studies in natural populations, no evidence that inadequate pollination drove the evolution of autonomy or selfing potential. So no evidence ecological factors are driving mating system evolution. However, autonomy plus pollen limitation together drive outcrossing rate. That's what it says. Okay, so putting all this together, what I have showed you is that selfing ability increases towards range edges and with colonization distance. Secondly, that there is a signature of post-glacial range expansion in the genetics of populations, with range edge populations, those furthest from the glacial refugia, having less genetic variation and more drift load. So we might predict that this reduction in genetic variation would favor the evolution of selfing simply by reducing the cost of inbreeding. However, we did not find a reduced cost of inbreeding in these range edge populations. Inbreeding depression. Inbreeding depression varied, but did not have this geographic signature to it. Then we looked to ecological factors to see how they might, to see whether they have given rise to this variation in mating system. And again, we found no evidence that contemporary ecological factors, in particular levels of pollinator activity, drive mating system evolution. So, in fact, we found kind of the opposite. That is, mating system seems to drive ecological relationships. Okay, so you might expect that at this point we are pretty discouraged. We have done a lot of work and evaluated a number of hypotheses, and yet we are no closer to answering the question why there's greater selfing further from the refugia than we were when we started. Period. That's where things stand now. So our question is, so we're stuck with this question. And our current working hypothesis, and one that I wish we had tested with these other ones, is that selfers are simply better colonizers, right? That this process of colonization favors individuals that can self. Why? Well, because if you think about it, a new colonist may be the only individual present. It may be that there's some 2 or 3 individuals that arrive at the same location, and any individual that is slightly better at selfing is going to make, under those conditions, or selfing without a pollinator, I should say, under those conditions is going to make more seeds, have higher fitness. In the world of plants, this idea that selfers are better colonists is referred to as Baker's Law. So this is something that we are currently testing. My graduate student, Hannah Makowski, is taking populations that have high selfing potential and populations that have low selfing potential and planting these populations beyond the range edge to see, in fact, if having greater selfing rates makes you a better colonizer. In addition, she's looking downstream at inbreeding depression. You might be thinking that the answer to this is straightforward. Of course, the ability to self will make you a better colonizer. And this is where I, just to try to not overload you with details, skimped on some information about pollination in this system. I mentioned that 2 of the 3 pollinators I showed you eat pollen in addition to pollinating. And in some populations, they eat a lot of pollen. And the greater the frequency of the megachilid or the halictid pollinators, the greater the pollen limitation. So just because populations can self doesn't mean that reproductive assurance actually occurs in natural populations. And that's what she's evaluating with these beyond range studies. Okay, so stepping back, as evolutionary biologists, our go-to explanation to understand phenotypic variation is often to assume that it has been shaped by local ecological factors. And what I have shown you and what I've talked about today is that it is important to consider these contemporary factors in the context created by historic factors. And then another thing that's interesting about this result is that these sorts of historical factors are geographically broad, and many species share the same history. So what that suggests is that there may be groups of species that share the same genetic structuring of variation among populations, and that has led to some of the same evolutionary potential and evolutionary constraints that we find in Campanula americana. And then just last, before I finish, thinking about current warming climates and the range changes that are associated with these warming climates, what this suggests is, as we try to understand what these range changes mean, it's important to think about how they may affect the evolutionary potential of populations. And with that, I will stop, and I want to acknowledge people and funding sources that have helped with this work. Before I mention Matt Koski and Jeremiah Bush, and then many students that helped count and plant many, many seeds. And then finally, the National Science Foundation for supporting this work. Okay, so that's it. Thank you very much.
Thank you very much, Laura. Come back. Thanks you for this very interesting talk. So there's no question in the Q&R, but there's one in the conversation thing. Right.
Bruno Fadi, let's see if we can activate his microphone. Can we? Or can you?
I think yes.
Converser.
Okay.
Yes,
there
I
you
can,
go.
you can.
Ah, c'est bon.
Hello.
Yeah.
Hi. Yeah, I had a question about density. I don't think you talked about that, but in outcrossing conifers, selfing definitely increases with, well, actually when local density decreases, contrary to what I wrote. And there's apparently no signal with biogeography or colonization history, as you showed. So I was wondering if you tested this density effect on outcrossing in your species.
We did. We looked at density of natural populations and found that there was not a density. I mean, I have to say I haven't looked at this for a little while, but my recollection is that there was no density effect on outcrossing rate. But I have to say that I'm not very surprised about that, because I think, for example, relative to your conifers, these are, this species has a much more patchy distribution, and populations are local with with not a huge amount of variation in density, if that makes sense. So you tend to have maybe a large population has hundreds, but not, often maybe not more than 500 or so plants, and is geographically pretty constrained. So I think it's the patchy distribution.
Okay. Maybe while we wait for more questions, I had one myself. I found it quite surprising to see that there is no relationship between the autonomous selfing that you measured in the greenhouse and the outcrossing rate in the population. So I was wondering, what are the traits that can determine autonomous selfing? Is there some kind of distance length of the stele or something like this? And do you know, well, do you have an explanation to the fact that sometimes in the greenhouse they won't be able to self-fertilize, and in the field you measured really high selfing rates?
Yeah. So I think I have two answers. I'll try to remember them both, but just keep asking me questions if I don't. So I didn't mention, I just forgot to mention when I was on the showing about flowers and pollinators, the species is protandrous. Okay. So when the flowers open, they're in male phase. And actually, I'll show you a flower pictures. Might take me a while to get back to. Wonder if it's worth it. So, yes, here we go. So see this pink right here? They have secondary pollen presentation. So the pollen is deposited on the outer surface of the style. That's why pollen consumption is such an issue, because it's just sitting out there exposed to pollinators. It's held on by these pollen collecting hairs that are touch sensitive. So when the pollen collecting hairs are triggered, then the flower moves into female phase. So what this temporal separation of male and female phase does is it makes automatic selfing less likely, right? Because if pollinators are present, the pollen is going to get removed before it moves into female phase. The question then becomes, why, if pollen is limiting, I'm sorry, now I've forgotten your question. So you're surprised at the relationship between outcrossing rate. So if they have high autonomous selfing in the greenhouse, why aren't they selfing in nature? And the answer is because if pollinators are frequent, the pollen gets stripped and they can't self automatically, right? They receive outcross pollen under high pollinator conditions. For populations that do not self automatically in the greenhouse, you can find high selfing rates in the field. That is, I mean, we haven't done this study, but I'm sure it's gynogamous selfing. Because you can see just from this figure, many flowers can be open on a plant at one time. So so that's why autonomous selfing is just an estimator of the potential to self without a pollinator. If pollinators are present, it just changes the game. Does that?
Yes, it does. And that maybe a follow-up question would be, I suspect there will be quite a lot of variation from one year to the other in the outcrossing rates. And would you by any chance have some data about this? You measured it carefully once, but do you have maternal descent for more than one generation?
Yeah, so we have, I'm trying to remember about this. We have 2 years of data on pollen limitation. And pollen limitation did not differ statistically between 2 years. In fact, it was, for most populations, pretty tightly associated, suggesting that there is some existing pollinator community and the predictability is pretty much consistent across years. When we did outcrossing rates, I'm trying to think. Anyway, we only wound up with one outcrossing rate, and we wound up combining them for statistical power reasons. Just figuring out what the outcrossing rate was. I did not mention that, and I should have mentioned because it impacted all the genetics. The species is an autotetraploid, which just makes getting an outcrossing rate more demanding in terms of a sample size, because you're dealing with genotypes that have 4 alleles.
So you couldn't get an estimate of outcrossing rate from your RAD-seq data, for example? Yes.
Yeah, we did have an estimate of outcrossing rate for RAD-seq data, but that was only 1 year.
Okay.
So that didn't give us more information. The information we got, the F value from RAD-seq, was pretty much in line, but less variation than what we measured using progeny arrays.
Okay.
I had, if no one has other questions, I had some questions about the drift flow. I was really interested in this part. I was wondering, so the way you estimate this is really through measuring their absolute fitness in different parts of the range, if I understand well. So it's a matter of interpretation in a way, whether this is drift load or just other factors explaining this reduced absolute fitness. If you see what I mean, can you establish that this is drift load? I'm just wondering.
Okay, so so we measure drift load using heterosis, right? So
Sorry.
so using hybrid vigor or heterosis.
Okay.
Okay, so we took crosses between two populations, made hybrids, grew the hybrids and grew the parents, and measured hybrid vigor as the extent to which the hybrids performed better than expected mid-parent average.
With the idea this is caused by associative overdominance caused by different drift load in the parental population, something like this.
It can be dominance. It doesn't have to be over-dominance, right? Because if you have...
Yeah, of course, yeah. Yeah. I meant associative overdominance, but in fact, it's recessive mutation at different loci.
That's, yes, that's right. That's
All
right. And so, sorry, so we also looked at just performance, like Yvonne Willey has done some really nice work where she has just shown a decline in performance with colonization distance. And we found that in our data, so a reduction in fitness with greater colonization distance leads to reduced fitness. But I have shied away from showing that data based on reviewer response, because reviewers say, okay, that reduction in fitness could simply be due to local adaptation, right? Your greenhouse is located in sort of the more southern part of the distribution. So perhaps as you're getting further from your greenhouse, you know,
this was sort of where I was heading. So I think I find it much more convincing what you explained about the hybrid vigor in terms of drift load, at least.
Right, because then you are using your parents grown in that location to standardize your expectations. Yeah. So, right, because otherwise getting away from local adaptation is almost impossible.
Yeah, so that was exactly my question. So I think you answered it well. But then I had a second question that was a bit more, I guess, well, I mean, you could say that if there is such drift load, or if there is drift load, isn't one possibility that the evolution of selfing is just part of this, or selfing ability is just part of this drift load? There's relaxed selection on avoiding to self or something like this. I don't know. I mean, it's just playing devil's advocate a bit.
So one question I have, so in years past, I did artificial selection on flowering time. And the amount of response I got in 3 generations of selection was huge. So I select for 3 generations, can separate flowering times of high and low lines or early and late lines by almost 3 weeks. All right. So lots of genetic variation out in these populations, right? And so the question is, if somehow, so the question is, why do we still find right now this geographic variation in autonomy, or the ability to self, if it developed as a product of range expansion and is not relevant now? This is not quite the answer to your question, maybe going there, right? Because what I've shown is that now there's no relationship between outcrossing rate, pollen limitation, and the ability to self. So why is this geographic pattern still maintained over time?
But what I was proposing is perhaps it's just some, non-adaptive pattern that's caused by fixation of mutations by drift, or if mutation tends to disrupt this trait, and then there's lots of drift.
I understand it, but the reason I don't I hear what you're saying,
Mhm.
and in absence of other explanatory values, I'm looking for something. But the reason I have a hard time accepting that is that then I can say, okay, well, how do they do this automatic selfing, right? Why is it that some populations self and others don't? If I showed you the two flowers, they look exactly the same. What I know is that the populations with high selfing ability have a bit less protandry, and it appears, we know that when visited by pollinators, they hold their pollen for a bit longer. We think maybe either the pollen collecting hairs are less touch sensitive, they're longer, there's something, we're trying to figure this out now. But that ability to self automatically has, you know, traits that underlie it, things that give it the ability to self. So these are acted on. It's very hard to believe that this kind of complicated trait is just a product of drift.
Yeah, it would be sort of the reverse. You would sort of tend to lose this trait by drift rather than gaining it.
Exactly.
Yeah.
Exactly. And so therefore, so that makes me feel like, okay,
Yeah.
I tried, I explored, and there's something out there causing this pattern, and I don't know what it is yet. And if you have an idea for what it is.
But it's stimulating to think about these things. So there's another question by Bruno Fadi. So I don't know if Lorraine can turn this mic on. I don't know how to do this. I can't do this, I think.
There you go. Bruno?
Thanks. Yeah. Sorry to go back to a previous to the previous part of the discussion, but I'm not sure I understood well. When you did your test for hybrid vigor, you did it in just one location in one greenhouse or in several greenhouses?
One greenhouse.
So going back to this local adaptation thing, which local climate did this greenhouse mimic, or did it mimic any local climate?
Well, the greenhouse was, of course, where my university is, which is, I mean, we set the greenhouse conditions to be those for high growth. So if climate is important in any way, my guess is it probably has to do with day length cues. That, you know, even if you put lights on in the greenhouse, the plants still receive day length cues. So it is, the greenhouse is slightly more southern, but we grew both of the parent populations in the greenhouse.
Right, but I'm wondering if you had done that in different types of climates or light intensities or so on, maybe the outcome would have been different. For example, the local population, the parent coming from the local population might have shown a increased vigor compared to the hybrid.
So not having done that, I don't know. What I do know is that the hybrids on average outperformed both parents. I mean, it just looks like heterosis. But I can't, I mean, I only did this in one location because populations were coming from everywhere.
Yeah.
But I didn't do it outside. I mean, it's just your sort of generic greenhouse environment.
But you had, I guess, different crosses between different populations from different parts of the range, right?
Yep. Yep. And in the crosses were done not randomly, but we tended to cross populations. Well, all right. So this is where life gets complicated. When I started doing this work, it was before we had the RAD-seq data. The RAD-seq data that I talked about just came out actually maybe this week or last week in molecular ecology. So we didn't have that at this point. We thought the refugia were in the south. We didn't know that the refugia, and right, because I showed that there was that white haplotype along the southern edge of the continent and in the southern Appalachians. we thought this was the refugia, and this was just kind of some leftover. We didn't really know what it was. It turns out they're both refugia. Probably these ones in the deep south are very old refugia from a previous glaciation event, and the most recent refugia is this one that the nuclear DNA show. Okay, so when we designed the experiment, we thought the refugia were down here. So we tended to cross southern populations that were in more southern latitudes, mid-latitudes, and north latitudes to each other, with the idea of crossing longer colonization distances together and shorter colonization distances together. The truth is, because the refugia was elsewhere, it mixed it up some, and so it turned out to be, I think, probably a better design than what we had initially planned.
All right. I think we've way over an hour now.
Yes.
So we perhaps should stop. I'll let Lorraine finish it.
Yeah, I will thank you again, Laura, for this great talk. This was registered, so in case anyone wants to watch it again, we can send the link. Perhaps we'll post it on the LabX website also. Yes, well, I think that's all. If anyone wants to chat with Laura, I think there's possibilities to meet on Monday, so just email us and we'll arrange something. And yes, I think with that, I'll wish you all a good weekend, and we'll meet next week. I can't remember if it's in the morning or the evening. Watch your Evolmomp emails. So thank you, everyone.
All right. Bye-bye.
Thank you so
Bye-bye.
much. Thank you for all the questions. And if you want to talk more, I'm happy to.